Hello, thank you for the invitation. And I just present me. I'm a professor in oncohematology, Annelise Bennaceur Griscelli from Paris-Saclay University. And I am a director of CiTHERA, the Center of iPS Cell Therapy. And my work is focused on clinical translation of iPS technology for cancer immunotherapy and genomic modeling of hematopoietic malignancies. So today I will present our very preliminary results using optical genome mapping in GMP-compliant iPS cell line. And so I will just explain you that it is well now known that pluripotent stem cells exhibit a high number of genomic and epigenetic diversity. Mostly in different cases during the derivation of human embryonic stem cell, but also during reprogramming of somatic cells and during iterative passage. So now the map of these recurrent abnormalities has been well established. So these alterations include aneuploidies and recurrent CNVs with specific hotspot region on chromosome 1, 17, 20, 21, 18, and X, as well as various non-recurrent alterations. In addition, it has been also well established SNV encoding region of cancer-related genes. Among them, the most common mutation involves the TP53 in 20% of the research-grade iPS cell line, and more recently, a B-core mutation, which is an epigenetic regulator involved in hematopoietic malignancy, has been well reported in a high repository of iPS cell line. 26 of iPS cell line can derive from blood harbor this mutation, and as well mutation in mitochondrial DNA. So what is very important now is to explain and to explore the functional impact of these CNVs for therapeutic and application, and as well research. So it has been reported that specific recurrent CNV can alter the phenotype, maturity, and functionality of differentiated cells type, such as the gain of chromosome 17 Q could accelerate the neuroectodermal differentiation. For example, the gain of chromosome 1 Q can alter the cardiomyocyte differentiation efficiency. And the 20q11.21 CNV can also alter the neuroectodermal differentiation efficacy and the hematopoietic differentiation. One of the critical main question remains to the risk of tumoral transformation in a clinical setting, and the question is whether cells harboring these first genetic variants are they predisposed to secondary oncogenic events, and so by acquisition specific hallmarks of transformation during the behavior of the cells in the body of the patient. So the key challenges is to ensure this genomic stability and safety of therapeutic products. So it requires multiple scale monitoring approaches to select and to follow the iPS clone and control the final product through the manufacturing workflow. We need methodology reducing their prevalence during the programming and expansion, and to prevent and eliminate these unstable clone in a master cell bank, but also in working cell bank and the final product. And importantly is to assess the functional significance of these variants by studies the downstream effect of detected genomic variation, and mostly in three critical safety pillars: the differentiation, potency, the tumorogenicity, and immunogenicity. So it's now well established that the ICCR guidelines and the FDA draft guidelines for industry mandate a periodic genomic monitoring of continuous cell lines in master cell bank, in working cell bank, and as well genome edited cells. So the non-directed tests are the whole genome sequencing, but also sensitive methods detecting structural aberration not visible by a conventional karyotype. So in our lab, we used the genetic analysis kits based on qPCR, quantitative PCR designed for rapid screening of hotspot region on chromosome 1, 4, 8, 10, 12, 17, 18, 20, and X. But what it was important now is to compare this technology with optimal genomic mapping, and we use two distinct cohorts of iPS cells. So we explored the utility of OGM during iPS manufacturing, and we evaluate the technology in two distinct cohorts of iPS cells. The first was in a research-grade disease-specific iPS cell line from different donors. And the second one, which is important to have this because we use it as a baseline control, because in this cell line we have a genetic signature related to underlying pathology, and so we expected to see the well-known structural variants. The second one is a clinical grade iPS cell line, and we analyze seven iPS cell lines derived from cord blood unique and leukapheresis of normal samples. All of them were produced in our clean room facility at CiTHERA under GMP-compliant manufacturing conditions. That has been all of them has been produced at CiTHERA in the clean room facility of CiTHERA. So the study design was performed to ask at least 5 questions. The first is the impact of reprogramming technology using integration-free Sendai virus and mRNA on the same donor, but as well the starting somatic cells. Secondly, the clonal heterogeneity. So we push the question: do sister iPS clones derived from the same donor differ in their genetic integrity? The culture condition is also important to be explored and to see if you have an influence of different media and feeder-free culture system on the genomic integrity, as well as the off-target effect of gene editing, and at the end, the clonal dynamics and scale-up expansion. And we explored 2 types of expansion culture, 2D and 3D bioreactors system. So we use at least 1 million of cells per sample, and we use a rare variant analyze pipeline for our analysis. So first, we wanted to validate the OGM sensitivity and accuracy to detect the well-known 20q 11.21 micro duplication. So we identified this first recurrent duplication in human embryonic stem cell in 2008. As you can see here by fish, we have 3 signals of the chromosome 20. And it has been later identified in iPS cell line. And this locus is very important because it has been observed in a patient that it can involve in human malignancy with very bad prognosis. And mostly this duplication was identified in colorectal carcinoma. So what here you can see an example of what we have found in a research-grade iPS cell from a sample of a CML chronic myeloid leukemia, and you can see the translocation first, the translocation of 922 with BCR Belson, involving BCR Belson, but as well an association with a 20q11.21 duplication with a precise breakpoint, and we can have unlike the PCR-based kits that only confirm the presence of the gain, the OGM could, of course, define the exact architecture of the duplication and as well the overlapping genes and we can see here the affected among these genes, the BCL2, which is an anti-apoptotic genes, improving the survival of the iPS during the culture. So the next step that we want to evaluate the immediate genomic impact of the reprogramming process. So we compare the iPS clone with the starting parental cells, and we can see that mostly of the alteration gene from the donor are also observed in the iPS cell line, but we have also multiple structural variants, CNVs, highly expressed in the iPS cells that had been acquired during the reprogramming phase, that were completely absent in the parental cells. So that is important to confirm that the viral and non-integrated viral virus can as well modify the genomic integrity of the cell during the reprogramming. Another critical variable that we look at it was the origin of the starting somatic material, and so we found similar. So we start from an iPS. From iPS derived from T cells or from CD34 positive cells. And we can see a similar viral mediated cellular response with the same type of CNVs between both of iPS, whatever the type of cells from the donor cells at the beginning. So another variable, which seems to be very important, is to explore the culture condition and mostly the culture microenvironment using extracellular matrix. So we compared iPS clones maintained in the same media across two distinct matrix types like vitronectin and laminin. So we found similar alteration between clones, except the lowest expression of CNVs in laminin conditions. And so it is one result, interesting results, very preliminary, indicated that laminin appears to reduce incidence of acquired CNV compared to vitronectin. So we are now expanding this study by additional iPS clones and passage, and to confirm the results and see if laminin can maintain a protective effect over extensive passages. Another one beyond the extracellular matrix, we examine also the media, culture media. And so we use, for example, here these experiments. So we use an iPS from a normal PBMC samples in a GMP media A on laminin during 10 days. And then we make a switch, we switch the cells from media A to media B, which is a research grade, very useful research grade media during 5 passage. And what we can see after 5 passage, we saw that the iPS cultured in RNA setting, we can find the translocation 774 after these 5 passages. So, and these results demonstrates clearly that switching media can act as a selective pressor and can rapidly alter the clonal composition of a culture. So in conclusion, unlike qPCR-based kits, OGM provide unbiased view of the entire genome, capturing non-recurrent rearrangements unique to specific donor lines, which is interesting because we can use this assay as a genome identity between cell lines. We can define the exact architecture of alteration and identify overlapping genes within the affected regions, which is important to explore, to have a comprehensive landscape of these different oncogene and gene suppressor gene during the culture. There is a high, I think there is a high utility of OGM in pluripotent stem cell manufacturing, as we can identify the first genetic hits and compromise the clone before investigating costly scale-up and differentiation process. We show also that the media and extracellular matrix are acting as a selective drivers of subclonal structural variants, and it allows to optimize the best culture conditions. So based on these results, preliminary results, I think that OGM is very useful and should be integrated to the mandatory regulatory tool kits for the qualification of qualification of master cell bank, working cell bank, and final cellular products. It will be very interesting to have these tools to identify the first genetic hits and compromise the clone before investing in costly scale-up and differentiation, as well as to ensure a novel cryptic alteration before to reach the patient, that has been to be managed before in the final product. But it still remains a different question. What are the variability among these different cells and sources in rate and type of instability regarding the donor age and the type of tissues and exposure history? And what is exactly the threshold to be used for all this mutation burden, and what are the acceptable for the therapeutic use? And for the clinical aspect, we need to harmonize regulatory practices with acceptable limits across labs and country regulatory agencies. So I would like as well to thank all my team and the teams in INSERM and CiTHERA, and especially engineer who help us to the manufacturing the IPS, Zara Kassem, Diana Sheker, Nufisa Oudreri, Theodore Latsis, and Hudson Bezera. So thank you so much, and open for questions.